How does your mind work? How does your brain work? What is memory? These questions have been puzzling humanity for a lot of time. Over this time, a number of sciences have emerged trying to provide answer. Since the middle of the last century though, a particular scientific discipline has been increasingly consulted: neuroscience. Neuroscience is the study of the nervous system. The nervous system includes the spinal cord, a network of sensory and motor cells throughout your body, and the brain. The brain is the most interesting thing in the universe and has been studied intensely. Neuroscientists have developed many different techniques and methods for this purpose. I will picture 3 of them in this video. They are quite rarely used, but when they are, the results are fascinating. Let me start with 2 methods from a specific branch. They are called invasive lesion studies. In these studies, areas of the brain are targeted to be intentionally lesioned to determine what function they perform. In other words, how would your behavior change if I were to destroy something in your brain? Depending on what would change, we could infer the function of the area involved. If you want to get rid of something small and don't want to exert yourself, most people simply turn on the vacuum cleaner. Believe it or not, neuroscientists are doing sometimes the same. There is a kind of a technique called aspiration method, in which a small tube is inserted into the brain. A vacuum is then created into the tube, which sucks out the underlying tissue. This is, for example, used for excising tumors, but also to gather information in neuroscience. Deep in your brain are 2 very important structures located. They are called amygdala and hippocampus. They are important for cognitive functions like emotional processing and spatial navigation. In the 1970s, neuroscientists wanted to figure out how these regions are important for memory as well. They performed studies with 12 monkeys which were divided into 4 groups. One group had only the amygdala removed, one group only the hippocampus, one group both. The last group, the control group, was not lesioned at all. Afterwards, their memory was tested. But how do you test the memory of monkeys? I mean, they cannot simply articulate whether they remember something or not. There's a simple design to solve this. First, you show the monkey a treat. Then you put a unique cover over it. You add a second cover that is marked differently. After blindfolding the monkey, you switch the covers. If the monkey then picks the cover with the treat underneath, his memory works. The scientists found that only the monkeys that had both the amygdala and the hippocampus removed were significantly less likely to find the treat. They concluded that both the amygdala and the hippocampus were crucial for building memory. The aspiration method has certain advantages and disadvantages. The technique allows for the complete removal of large brain structures. On the other hand, it's difficult to preserve adjacent tissue during the procedure, and this can be a problem. In the 1990s, the study was repeated. This time, scientists used a more precise version of the aspiration method than the previous study. And the results differed. They found that not only the amygdala and the hippocampus were crucial for memory, but the tissue around as well. Quote, they found that lesions of the hippocampus and the amygdala produced the most severe memory deficits only when the cortex surrounding these regions was also lesioned. Besides vacuuming, there's another way to make sure that parts of the brain become dysfunctional: cooling. When brain matter is cooled to a certain temperature, the neurons stop functioning. This can be seen in this graphic. The neuron is active at body temperature. If it's cooled down to 8 degrees Celsius, it stops its activity. If it's cooled back to normal temperature, its activity returns to normal within 5 minutes. This phenomenon can now be used for neuroscientific research. A cooling device is implanted into the brain. If it's turned on, it prevents the surrounding tissue from working properly. We can then infer the function of the area involved. You can see such a cooling device here. The loop contains cooling fluid, which lowers the temperature of the area around. If it's implemented, it looks like this. In the past, this technique was, for example, used to study the auditory parts of cat brains. To do this, cooling devices were implanted in different places of the brain, and then the cat's reaction to a sound was tested at different angles. This method is especially effective for inactivating larger areas of the cortex. However, it requires direct access to the tissue, which makes it less suitable for reaching deeper brain structures. At this point, it's important to say that the methods discussed are not performed on humans. They are instead performed on animals. In recent years, the use of animals in neuroscience has developed to increasingly implement ethical standards and guidelines to ensure animal welfare and to avoid the use of animals where possible. In this video, I merely want to inform. I do not want to discuss the moral problems arising with experimenting on animals. If you're interested in that, I've put some links in the description. However, the last method I want to show you can be easily applied on humans. And actually, it's kind of fun. Neurons communicate via electrical signals. When this electrical communication is exposed to a magnetic field, it is influenced by it. This fact is used by the so-called transcranial magnetic stimulation, or TMS for short. A point source magnetic field is generated via 2 coils and then focused on a specific brain region. The level of brain activity can be increased or decreased based on the specific parameters of stimulation. You can do all kinds of stuff with this method. If you target, for example, the area in your brain responsible for color perception, you briefly see the world in black and white. If the TMS is turned off, the colors are returning to your vision. Another example. The area in your brain responsible for initiating motion is called the motor cortex. In this video you can see the TMS interfering with it, resulting in a deficit of motion. Strange, isn't it? I hope I was able to give you a little insight into a few neuroscientific methods. There are many more that have answered significant questions but have raised even more. The scientific journey continues. And this is great.